All right. Next speaker in the program is Professor Saumitra Das. He is currently acting as Director of National Institute of Biomedical Genomics, but he belongs to Microbiology and Cell Biology Department, where we are also. And from now on until the lunch break, I'll request Professor Deepshikha to come in and moderate the sessions. So, Professor Deepshikha, please share your audio and video.
Yeah.
Thank you. Thanks for joining. Thanks for making time for this. Professor Das, can you share your screen, please?
Yes, I have shared the screen. Can you see it?
Yes, I'll set it live now, and you can go from here. Yeah. And Professor Deepshikha, please take hold of the time and questions. Thank you so much.
Welcome, Professor Saumitra, and we are looking forward to hear your talk. Professor Saumitra Das is currently the Director of National Institute of Biomedical Genomics in Kalyani, and our very dear colleague from Department of Microbiology and Cell Biology, Professor at the Department of Microbiology and Cell Biology. And his decade work on the hepatitis virus have really added a lot of interest and wonderful work that he has done, and he's continuing to do so. And today we are going to hear from him about the SARS-CoV-2 genome sequencing, the variations and implications. Professor Saumitra, please.
All right. Thank you, Deepshikha. It's always a pleasure to either play in the home ground or play for the home team. So, and it's always comfortable when you come for the second down, because the subject is already introduced and you know the pitch and you know the spin, so it's relatively easy to play. So as Shashank and Deepshikha both mentioned, that my head and heart is now separated. Essentially, I am at the National Institute of Biomedical Genomics since 2018 September, and now ready to come back where my heart is at the Indian Institute of Science, where my laboratory is continuing for last 22 years, since 1998, I guess, yes. And we have a full-fledged lab going on, working primarily on hepatitis C virus, but actually our research interest spans for other RNA viruses, something like Coxsackie viruses. Lately, we started working on dengue viruses, and very recently we tried our hands in the COVID-19 as well, in collaborations with the group at the National Institute of Biomedical Genomics. Now, see, as I told you that already you have heard from the first two talks from Sumit and Yuman actually about the coronaviruses. So I will more focus on essentially the biology of the viruses in the beginning, and then I will talk about the genomics and try to understand the implications of the genetic variations in the biology of the viruses, which actually is central for the pathogenesis of this particular or the disease severity that we are facing. Now, as it's already told that the coronaviruses are actually RNA viruses, and in our laboratory is also interested and have been working on the different RNA viruses. As I told you, this RNA viruses, they actually goes inside the cells by the receptor-mediated endocytosis. In this case, the ACE2 receptor is being used, and then the virus goes into the endosome. So there is an endosomal fusion, and after that, the virus actually uncoats, and the viral RNA starts translating. Now, in the translation also, there are two unique things that we have learned from coronaviruses. One is essentially they are translated into two different polyproteins. One is the polyprotein 1A, and the second one is the polyprotein, which is actually the combinations of A and the B part, called polyprotein 1AB. And that is actually done by the ribosomal frame shifting. And that's also something very interesting about this particular virus. And then the non-structural protein and the structural proteins are made Then they assemble in the translation and the replication complex, which is usually in a double-layered membrane vesicles. And there now they replicate from positive to negative, and some of these negative strands are used for making more and more positive strand. Then ultimately the viral proteins are synthesized enough along with the nucleocapsid protein. Then they assemble, and then the proteins are actually of interest are spike protein, the envelope protein, the membrane protein, and the nucleocapsid protein. And then ultimately they are actually covered by these smooth walled vesicles, which actually protects them to some extent, and then ultimately they releases out. Now all these steps are coordinated by several proteins, which are actually host proteins, and some of them are viral proteins. They are crosstalk between the viral protein, cellular protein, or the viral protein, or the cellular protein. And more importantly, there are some RNA and protein interactions that actually plays major role during the course of this viral infection inside the cells, in the infected cells. So there are factors like the viral and the host proteins in the viral replication complexes. An important and specific role of the host factors in the RNA replication and the translation is actually we are interested in to understand and highlight, because this is relatively a new area to understand more biology. We are also interested to identify the host factors that are key determinant for the viral pathology and the evolution. Essentially, as we believe that there is definitely a role for the changes in the viral genome, the genetic variations, but it is more important is the host determinant, how patient to patient the virus can actually take a hit away, or actually causes different extent of pathology, and that leads to the disease severity in patient to patient. So we are trying to understand what is largely the host determinants. Just to give you a recap on the genome organization, as I told you, this coronaviruses, it's an RNA viruses. They have a 5' cap structure and the 3' polyethyl, and in between there are multiple open reading frames. The what if 1A and 1B I already mentioned, or what if 1AB actually is made because of the ribosome frame shifting, and the proteins that are of interest for most of this work that is going on around the world are the spike protein, envelope protein, membrane protein, and the nucleocapsid protein. The genome codes for a single replicase gene, which occupies almost the 2/3 of the genome at the 5' end. But you know, the replicase gene in the RNA virus is actually, it's a major player. They're not a good student. They keep on doing mistakes while copying. And that's how we actually get more and more trouble from the RNA viruses, because of this error-prone replication by these RNA-dependent RNA polymers. Now, after the replicase protein, they are followed by structural proteins. And the structural proteins are, as I told you, are the spike, envelope, membrane, and the nucleocapsid, occupying the remaining 1/3 genome. The gene order is first is the replicase, then this spike, envelope, membrane, and nucleocapsid. Then there is a 3' end, 3' UTR as well. The complete synthesis of the replicase polyprotein requires the ribosome frame shifting, as I have been referring. The replicase assists the synthesis of the genomic RNA as well as multiple subgenomic RNA, and the non-structural proteins are synthesized from the subgenomic RNAs. N protein is the nucleocapsid protein, which binds the genomic RNA to form the viral capsids. So any mutations in the N protein will have some implications on the viral capsid interactions and with the RNA, and also the assembly of the viruses. The spike, envelope, and the membrane are wherein the spike protein is the major envelope glycoprotein, which is involved in the fusion of the viral particle with the host cell membrane. This is already highlighted that, you know, how we actually get this jolt from the RNA viruses because of their antigenic shift and antigenic drift, and sometimes they ended up with a recombination, and there are changes in the receptors, in the ligands, and that actually alters the receptors and the ligand, the structure, and finally their affinity and which has a tremendous impact on the infectivity of the virus. As you see from a natural host, as Sumit was showing, and then also my earlier speaker has already highlighted, how the intermediate host can play a role to get into the human host. And in this perturbation of the structure is something leads because of the mutations that the virus acquires in the intermediate host. Now, with this background, and actually, around March and April, when we actually started really looking at the pandemic and then how we can survive under this situation, there was a request from the Ministry of Science and Technology, none other than our Minister, Dr. Harshavardhanji. He said, why don't you just try to sample different parts of the virus isolates from different parts of India, because India is as diverse with the genetic determinant compared to other countries. There is a lot of scope to understand how it can actually implicate to different populations, and try to understand why and suddenly in Mumbai and Delhi or Calcutta are behaving differently to this particular pandemic. So with that, the Deputy Secretary, Dr. Renu Swarup, actually asked me to form a group as an initiative of the DBT Autonomous Institutes and come up with a consortium who can actually sequence in a span of 3 months the 1,000 genomes of this SARS-CoV-2 RNA collected patient samples from different parts. So in my group, what I did actually, we have actually Professor Arindam Maitro spearheaded the group in NIBMG along with me and Nidan. And then we had this IISc Bhubaneswar, Ajay Paridar's group. CDFD at that time, Debashish Mitra was actually the director, and now actually Thangaraj taken over with this Shin Dalal and Murli Vasham, where in NCCS, Manoj Bhatt and his group, and in STEM, NCBS, and they have also initially was with IISc, and later on I got samples directly from IISc, and that is a cluster where we collected samples from Apurva Sarin and Satyajit Mayar, helped us to get samples from the south, particularly in the Karnataka area. So they were the participating institutes. So the PanIndia consortium launched the program in April 27, as I told you, to get a countrywide landscape of the variations in the SARS-CoV-2 RNA genome. As I mentioned, that it was coordinated by the National Institute of Biomedical Genomics and 4 other national clusters, including the Bhubaneswar, Hyderabad, Bengaluru, and Pune, which have actively participated in sequencing and the analysis. And there are other collaborating institutes. There was ICMR, NICET in Kolkata, IPGMER, Kolkata, of course, IISc Bangalore, along with AIMS Uttarakhand, then Maulana Azad in Medical College in Delhi, THSTI Faridabad, then GMC Aurangabad, this Wardha, Mahatma Gandhi Institute of Medical Sciences, RMRC Bhubaneswar, AFMC Pune. RMRC Bhubaneswar was actually along with the IISc Bhubaneswar, and then AFMC Pune, the BJ Medical College in Pune. All these hospitals were actually coordinated, and we were NIB-UNG was actually taking the most of this lead to interact with them and collect these patient samples directly. From there to the RNA was transported in to NIBMG for sequencing, and some of the places like in Bhubaneswar, ILS, CDFD, or in INSTEM, NCBS, and NCCS, they have the sequencing machinery, so they could sequence themselves. But the rest of the samples were all collected in to NIBMG to sequence and then get these things down. So what we had it in mind to sequence 1,000 viral genome from samples collected from different zones within India, then try to understand the molecular phylogeny of the virus, which will indicate the track, how the virus is evolving in India. Remember, this was conceptualized in April. That time we didn't have a very clear picture. Now, obviously, the situation is much more stable and clearer. And then try to have the sequence and correlations with the variations in the viral RNA to the disease severity and the transmission efficiency, ultimately leading to identification of the strains which are associated with the enhanced pathogenicity. So this was actually the short-term goal, because we were supposed to deliver within 3 months. And the long-term goal obviously is more of basic research interest for us, is to identify the host genetic polymorphism that either confers the susceptibility or the protection from the viral infection. And then second is it possible that we have a signature to talk about the viral and the host genetic determinant that decides on the disease severity. So as I told you, so these places in yellow are actually the places roughly we have collected samples all over India we tried to cover, except the Northeast was actually still a struggle to transport anything from the Northeast to here because of the aircraft was actually not taking up patient samples, and that's why we couldn't get it. So the idea was to collect this nasal or oropharyngeal or the nasal swabs or the bell or the autopsy samples, and then try to get the viral RNA isolated from there and sequence them, then try to track the outbreak, the viral evolution, the host genomic correlates, and then try to understand the susceptibility and the resistance. And the patient data were actually shared along with us, and this is essentially a challenge. You can understand, as I told you, that to get patient information from all different 10 states, it's not so easy, but we tried to get as much as we can, but we had definitely more handle on the patient samples which we collected at NIBMG directly. So those 466 samples, we had much more intense analysis, as I'm going to show you later. But overall, we could actually at least share the sequences and try to understand the phylogenetic time tree. So this shows that how it's actually started from January 2020, and then slowly at the end of January, then February, then March, then April, then May, and finally in June 17th. These are the collection times. And as you see here, all of them, although originally it started with the O type and the A2 type and the A3 type, and then there were 19B samples like the B1 and B4, but later on it actually settles down to 20A, 20B, and 20C. Now 20A, 20B, 20C are actually new nomenclature. Earlier we used to call this as the A2A, which is predominant along with the few parental, the haplotype, as I mentioned, which originated from Wuhan, is the 19A and B. And then we found actually in India, this is very interesting, multiple lineages were introduced which are evolving over time. So this is essentially the thing that we could actually get the pan-India picture in different time points. Starting in the March, you can see most of it was actually by the A2A, but there was a considerable populations were actually infected still with the O type. And then this is the situation in April, this is May, but by June you could see A2A has emerged as the predominant haplotype all over India. And the temporal haplotype diversities and the landscape appears to be very similar, like the time frame where actually overshadowed everything, A2A overshadowed everything by June in every parts of the country. And we divided the samples into eastern zone, southern zone, western zone, and the northern zone. Eastern zone, the samples were collected mostly from the West Bengal and Odisha, and a little bit from Jharkhand. And then in south, we collected from, as I told you, from Bangalore, the IISc Bangalore, then INSTEM and CBS, they also collected somewhere else from Karnataka. Then Telangana was actually collected from the Hyderabad center in CDFD. So that comprises of the south. And then west actually was mostly collected by Pune center and from Mumbai and the other areas. And the Uttarakhand, Rishikesh, Haryana, and Delhi central, we actually collected by the directly from NIBMG by coordinating with the hospitals. Now, as you see here, the haplotype diversities peaked between March to May, an early part of the outbreak. By June, it all settles down to 20A, 20B, and then between the east, west, north, south, there is some differences, and you can see some similarity between the how the the haplotype diversities emerged in the West versus in contrast in the South. You can see it actually, the differences, these are actually different genotypes, I mean, different haplotypes are the O type, then B2, A3, A2, and A2A. A2A is actually, you can actually see that from April, this is the situation in May, this is the situation in June. But if you really compare with the South, South is actually, we didn't have considerable populations of the B2 and the A3. So these are the actually, if you really go detail into it, the haplotype distribution region-wise, you can see the northern region and the eastern region somehow behave similarly. You can see the 20A, 20B, and 19A and 19B is actually some, the way they have actually emerged. It's very similar graph pattern compared to West and the South. Within the haplotype, the states appear to have different proportion. You can see at a different time, the proportions of the 20A, which is the A2A, is actually, varied between the states, and we really don't know whether that has really contributed to the fatal cases or even much more disease severity or much more transmissibility at those time points in those parts of the those states. So this is an haplotype network that IISc Bhubaneswar has come up with the sequences that they have got it, and along with the others, that it says the haplotype nodes with majority of the genome from West Bengal, Orissa, and a small percentage of the samples belonging to Uttarakhand. Geographically, Orissa and West Bengal, they share border. So it is understandably that there are actually people moving forward, and particularly the Bengalis are known to be a good tourist, and they keep visiting Puri, which is a religious place in Orissa. So actually we could see there are, because of the border sharing, we could see there are the haplotype sharing as well. Two haplotypes might be because of the high interstate traveling. Maharashtra, Delhi, Haryana, and Uttarakhand grouped together with 2 to 4 single nucleotide variations, suggesting the infection might have spread in a short duration of time because of the travel and then people homecoming. There is a portion of the samples from Haryana and Karnataka sharing same parent haplotype, representing the possible transmission by the migrations. And all these data were actually very, very comfortable with the negative estimate of the Kajima's score. And if you really try to understand that where from we got this virus and when did we get it, we actually did that exercise as well from the 1000 genome kitty that we got it. And actually the origins started, you can see, from Brazil, New Zealand, China, Oman, United Kingdom, Saudi Arabia, Italy, Greece, and Switzerland. You can see the 19A and 19B mostly came from China, and 20A, 20B, and 20C we got it from UK, Italy, and Saudi Arabia. There is one observation is that 20A came from Italy and Saudi Arabia in all Indian region, but in case of Eastern India, 20A came additionally from the UK and the Switzerland. It all depends on where our actually students were there at the time. They tried to come back. Parents wanted to come back to India, and somehow, unfortunately, they carried it, as actually was mentioned in my earlier talk, that there were a lot of carriers which we couldn't really identify them, and they somehow skipped the airport screening and actually spreaded the disease. The 20B was introduced mostly from the UK in all regions. Additionally, in the Western India, from Brazil and Southern India, introduced from the Italy and the Greeks. Now, one thing was actually we were facing that, we have been talking about that the A2A strain is actually overshadowing all other haplotypes, but an A2A is actually known to have much more susceptibility. But does it have anything to do with the infection rate or the virus titer? Actually, this we found something interesting, and I'm just sharing it. Is actually we found some marginally significant association of the sequences belonging to 20A, B, or C with a lower CT value. suggesting the virus titer was marginally high compared to the clade 19, the clade 20 we have done observation. Now, one cycle difference in the CT value is equivalent to twofold difference in the viral load. But again, it's actually, we should just increase this number of samples again to have our conclusions out of these observations. But it looks like that there is some differences in the CT value in the A2A versus the other strains that we were facing before June. Now, then we try to map all the mutations that we have got it from this 1000 genome sequencing and try to understand and make sense out of that. So what we try to do is to locate the non-synonymous mutations and the synonymous mutations, and we actually found that the most of these mutations, the non-synonymous mutations, are actually spreaded, but they are mostly in the N-terminal domain, and some are here, and but the synonymous mutations are actually again clustered mostly in the receptor binding domain and the SD2 domain within this span. Now, what is interesting then that we did find a lot of mutations, genetic variations, but when we actually looked at the table and their occurrences and tried to translate it into terms of percentage, they really didn't stand out. You can only point out to the mutations that is well talked about, that's D614G, that we found out of 466 that we collected in IBMG, of that 371 actually showed, which is 79.61% is of the whole all the 466 genome that we analyzed for this particular purpose. And the next one was actually 294 mutations in this spike, which was actually found the 10%. But the rest were actually either less than 3% or 2%, and some of them are in the ranges of 2 to 3% variations in terms of populations. So we really don't know that the diverse scenario that we are seeing are because of the genetic variation that we have faced, or is mostly due to the genetic determinant of the patient from the different states, and they are definitely the immune getup and the other setup is quite different compared, if you really compare northeast, southwest. So essentially, we are boiling down to more onto the genetic determinant that actually caused the different ranges of disease severity and mortality for this particular pandemic. Now, highlighting the mutations that we found in the nucleocapsid, again, these are observations. We found there is something very interesting, is the 203 and 204 mutations. This is coming out around 42%. Now, this results is done in July, but actually we published in May, I think it came out in June, that reported this mutation that was actually came out a very pilot study. We actually, Arindam could collect 9 or 8 sequences from Calcutta area, and we just did a pilot study to see whether are we seeing any mutations. We could figure it out, the D614G mutations at that time we reported, and we also found there's 203, 204 mutations in the nucleocapsid mutations. But it looks like this might have something to do with the virus fitness. We'll probably actually looking into it. And RNA-dependent RNA polymerase, we did actually found some mutations like P323L, and the other mutations we have already published it. Side by side, we were actually, we were also trying to understand, is it possible that we can find out some kind of genetic determinant information from the symptomatic and the asymptomatic individuals. Again, this is a very short and pilot study where we try to see that the RNA that we were isolated from the ball, actually the bronchoalveolar lavage actually also had some cellular RNA. We tried to sequence, because we did not do a PCR based sequencing, that time we were actually doing the total sequencing. So we had an access to the genomic RNA sequences and then actually try to see the genetic differences in there and what all genes are actually up and down in those. And we did find, this is again a very, very preliminary data, but I just wanted to share with you. But we are actually trying to work deep into it and then find out there is any meaning to that. that the HLA-A genotype associated, and actually interestingly the genes that we have shortlisted which showed differences between symptomatic versus asymptomatic are already some of them reported. So that gives us a strength, that our confidence that our assays are actually going in the right track, but definitely we need to address this more in the biology and try to understand how these genes can actually contribute to the disease pathogenesis. Now, this is something I was referring that we actually did this study, pilot study in May and published in June. It was actually almost within 2 weeks, the Journal of Bioscience could review and then publish it and put it into the public domain. That is a very, very fast job by Indian Academy of Science. This is the study that we did it with Indian Institute of Science. You can see our popular face Srinivasan, and my student Harsha is here, along with the our NICED people, that National Institute for Cholera and Enteric Disease in Kolkata, who has given the samples from Kolkata regions. And as I told you, this actually study showed that D614G is actually important. We also pointed out one more mutation, which is actually G1124V, and tried to understand what is the implications of these mutations. Now, I will just tell you a little bit about this spike protein. Now, the spike protein is actually a class 1 viral fusion protein, which is having a metastable pre-fusion state and a post-fusion state. And this is, yeah.
So Saumitra, you'll be winding up soon? Or?
Yeah, another 5 minutes.
Thank you.
Yeah. So this pre-fusion and the post-fusion actually is important because that determines the affinity towards this, and we could show that how this C614G mutation can bring in the flexibility of the SG domain, and that's probably somehow it has more susceptibility and affinity towards the SCE2. Similarly, as I pointed out, that R203 and 204, these mutations in the nucleocapsid, we showed that how it can changes the conformational flexibility between the neighboring amino acids, and that alters its virus assembly power and the packaging power, and we are trying to address that. Harsha actually showed that how these mutations in the nucleocapsid can actually alter some of the microRNA binding sites, and those microRNA bindings can be relevant in terms because their target genes are altered because of the mutations. Particularly, these mutations are results in the altered binding ability of the host microRNA to the viral RNA, and we expect that it might interfere with the host susceptibility. Now, all these things are actually observations and association studies. We wanted to validate in a virus-like particle, and that's what essentially we are doing in my lab at the Indian Institute of Science and also at NIBMG. We're trying to understand how those mutations in the context of a virus-like particle, if we incorporate this mutation, can we really show and demonstrate that how it affects the entry, how it affects the immunogenicity, and how it affects the pathogenesis. And the results are actually trickling in. Hopefully by end of this year, we will have some clear picture of all the mutations that we have observed so far in this genome analysis. Can we really incorporate into that this VLP and understand the biology and the consequences of these mutations in terms of disease severity? So as I told you, as on date that we have completed this analysis from 10 states, and we have shown that the A2A haplotype and with D614G mutation is nothing surprise. As of worldwide, it has been observed, and in India is also not different. What we want to do is essential, as I told you, that to validate this observation and try to understand the implications in the entry, immunogenicity, and pathogenesis in a VLP model. Try to understand the host genetic polymorphism and the how it confers a susceptibility or protection to the viral infection. We started getting results, but again, it's too preliminary to share with you at this moment, and investigate the viral and the host genetic determinant for the disease severity. So it's actually a big consortium. And as I told you, Arindam is actually young and then an energetic. He could actually bring in all these people together. As you see, there are 5 institutes involved, and there are several clinical collaborators or the people who have actually directly contributed the samples, including IISc Bangalore. All of these people are thanked, because without that we couldn't even achieve to sequence in 1,000 genome in 3 months' time. So I hope that these observations we can actually analyze it properly and make it really fruitful. And we want to expand this study to some more to the genetic determinant. Thank you, and I will be happy to answer questions. Thank you.
Thank you, Professor Saumitra Das, for giving us a deep insight into the pan-genome sequencing and also telling us about the different haplotypes as well as the mutations. So there are a few questions which we will take. The first question is by Shashank. His question is, what regions of the viral genomes are changing the most?
So the one feature of the virus is its attachment with the ACE2 receptor is changing. Now there are actually studies coming out that other than ACE2 there are co-receptors. We don't know whether the mutations in this spike will have some bearing with the crosstalk with the co-receptors. That actually we wanted to address it with the VLP model, and I think I will be in a better position to answer in December, not now.
Thank you, Saumitra. The same second question from Shashank again. Did the different states had first introduction of virus from different part of the world?
Right. Yes. Actually, it all depends on where from you got your old Indians coming back. So I think this is essentially the contributions of no offense, but a homecoming. So the students came back, their parents came back, and they brought in different strains from different countries. So it's all depends. Delhi is, cosmopolitan, and it has contributions from all over. Eastern India as also, I can see that we got it from Switzerland, which is very surprising. But mostly I think it was coming from UK. For most of the states, actually, we got contributions from UK returned people from non-resident Indians. Yes.
Oh. Thank you, Saumitra. And third question is from Professor Kumar Somsundaram, and his question is, can you translate the viral haplotype distribution to viral clades? Because this translation can give a meaning to the course of infection.
Right, we are actually trying. We need little more deeper analysis. As I told you, that analysis is now half done. We have done 50% of this, and once we get it, it is actually a good suggestions. We are also trying to see whether we can have a direct correlations, but it's difficult because, see, as I told you, to get all the informations from the directly the centers where from the samples are collected, and there is the struggle. Once we get it, I think we'll be able to establish that.
There is one question I'll take from the participant, and that question is that regarding the mutation, the D6142G, the question is how it would be, in your opinion, the most appropriate strategy to counter these emerging mutations?
So, I mean, definitely the vaccination is one thing. I didn't understand. I mean, you can't really, don't know, you don't have any control on the virus, how they will mutate, right? So yeah. Essentially, if you can somehow stop the transmission of the virus, that is one, either by protecting yourself from getting infected. The other one is protecting by the vaccine strategy. But specifically, you can't really stop a particular virus spread unless you have a neutralizing antibody which will selectively block only the D614G viruses. That's a possibility.
Thank you. Thank you, Professor Saumitra, for excellent